Thank you very much for this invitation. It was my pleasure to be here, and also my pleasure to give this first talk, as I understood now. Yeah, so I will be speaking about mainly my PhD work that is about corticokinematic coherence. So that is the coupling between hand kinematics and brain signals during fast repetitive hand movements. So hence the title of my talk, Insight into sensorimotor functions from magnetoencephalography and coherence analysis. So yeah, that will be mainly about magnetoencephalography. So let me just give you a brief introduction that I guess you already know that, but just to make sure that everybody knows. To control movements, it's mainly this area that you can see in green on this brain, so the primary motor cortex, that sends information to the periphery through the spinal cord, and then goes to contact muscle fibers. And then this information from muscle fibers and tactile information and everything, every sensory information comes back to the brain through this spinal cord to the area that you can see in red. That's the primary somatosensory cortex. So those two brain areas are well arranged, so that there is a good matching between limb and the cortex. So for example, you can see there that the foot is located in the interhemispheric fissure. The mouth is located on the lateralmost part of the gyrus, and the hand occupies an intermediate position. Actually, it's not really well known what's encoded in this motor cortex, whether the neurons code for muscle activity or whether they code for a more advanced kinematics parameter. So whether they code, for example, some reaching movements or something more higher level. And those doubts come from recordings in the primate cortex that show that neurons in motor areas have their activity modulated by several kinematics parameters, such as direction, speed, acceleration, static position, or accuracy. So such kind of findings have been also demonstrated in humans, for example by Jerby, who used a visual motor task. So what he asked subjects to do was to compensate for unpredictable movements of a cube displayed on a screen. So subjects had to manipulate this trackball that you can see there. So the cube was kind of rotating left, right, and in all directions, and moving this, they could compensate it. So hence producing some displacement of the hand. And what he showed was that brain signals are coherent with hand velocity, mainly at movement frequency. So from 0 to 8 Hertz, there is a nice peak of coherence. I will come back later to in the method part. I will explain what is coherence and so on. And this corresponded to the main power of hand displacement. And the brain area responsible for this coherence phenomenon was mainly the primary motor cortex contralateral to a hand movement. So the aim of my PhD thesis was to better understand the neurophysiological mechanism or processes responsible for this coherence phenomenon. So first what we did was to try to reveal what's the brain network responsible for this coherence phenomenon, this coupling phenomenon, using some self-paced repetitive finger movements, so using natural movements instead of visual task. And then to evaluate whether this coupling is due to motor commands sent by the motor cortex or whether it's due to proprioceptive feedback to the sensory motor cortex. Because of course when you make movements, the proprioceptive information is very important to guide your movements. And that is quite well known. And then we also made use of this coupling phenomenon to probe mirror functions in the human brain, because we had at hand some phenomenon that could kind of quantify the coupling between one's brain activity and the kinematics, so why not investigating the kinematics of some other person's movements, and then probe mirror functions in the human brain. And then the second part of my PhD thesis aimed at translating all those findings into clinical applications to map the primary sensorimotor cortex. So we made use of this coupling between brain signals and kinematics to get some good information to map this primary sensorimotor cortex. And also test whether including several localizers of the sensorimotor cortex could improve the localization quantitatively and qualitatively. So let's proceed with methods. So you guys just acquired an NatMEG system some couple of months ago. So let me just review the basic principles of magnetoencephalography. So the idea is that when a brain area activates, there is some current that flows through the dendrites of neurons. And if those neurons are ordered well enough in the cortex, and it is usually the case, so the dendrites are perpendicular to the cortical sheet, then if the brain area activated is big enough, this will generate a macroscopic magnetic field there that can be picked up by magnetoencephalography. So the system you acquired had 306 channels that are placed all around the head of the participant, patient or subject. And so hence you can record quite accurately the activity from the whole cortex at the same time. And then have access to some topography of the magnetic field in response to tasks. But then what one usually wants is to know where the activity comes from in the brain. And this you can do using some standard dipole modeling. So that is estimating the source in the brain that explains the best the field pattern that you observe. Or you can use also some more advanced methods such as minimum norm estimate or beamformer, and then estimate the activity in a grid constructed on the brain. So in the studies I will present you, I've been mainly using this second approach. So I will just review briefly how to do this. So first, we have to define a grid, a source space on subject's brain. So here you can see a 5mm grid source space on the brain. And then we have to define the spatial relationship between those sources and the sensors, the NatMEG sensors. Once we know that, we can link the activity at the sources to the magnetic field they would produce by computing this famous lead field matrix. And this can be easily computed because it's the magnetic field that would be produced by a single dipole that would be placed wherever, while the other dipoles have no activity. And then once you know this lead field, you can try to find some kind of matrix that you invert the problem, kind of finding an inverse solution that, when applied to the magnetic field, yields the current distribution. So one solution is the beamformer that has been proposed by Van Veen. So this beamformer kind of estimates the activity at a given source by trying to minimizing the activity coming from all sources around. To analyze brain data with coherence analysis, so one typically needs to have NatMEG signals from one side and analyze its data with some reference signal. So the reference signal can be some muscle activity leading to cortex muscle coherence, or kinematic activity leading to cortico-kinematic coherence. And I will be mainly speaking about the second option. Coherence analysis gives rise to two parameters of high importance. This one is the cross-coelogram. So it's really handy because it has the spatial distribution of a magnetic field. So once you compute this cross-correlogram between the reference signal and MEG signals, you can find the source. You can do some dipole modeling on this spatial distribution and find the source responsible for this coherence phenomenon. Another parameter of interest is the coherence itself, which is a kind of generalization of correlation. So it gives a value of correlation for each frequency being afforded by your windowing, kind of. So values, when coherence is 0, it means there is no coupling between the brain and the other signal at this frequency. And when coherence is 1, it means there is perfect linear dependence between those two signals at this frequency. But coherence, you cannot do any source modeling with a spatial distribution of coherence. So if you want to know the spatial distribution of coherence in the brain, you have to first use this inverse solution I just presented in the slide before, and then compute coherence in the source space. And then once you do that, you can have access to coherence maps at the subject level, and then you can do whatever you want. You can use it as a T-map, like it's used in fMRI, and do some group analysis with your coherence maps. Just to get some further insight about what's coherence, I've here represented 3 pairs of signals, which are all non-correlated, as you can see here, because when this one is up, this one is 0, and it's always, correlation is always 0 between all those signals. The first pair has perfect coherence, whereas the last pair has no coherence. And that can be seen clearly here. So when this one is up, this one is always 0, and it's like they are in phase quadrature. Okay, they will always follow each other with some kind of jitter. Here, there are no phase relationship. For example, this one is up, this one is down, and if we follow, now they are better overlapping. They are even overlapping there, and then again in phase opposition. So this kind of cancel and destroys all the coherence. And here it's more like an intermediate position. For example, there is a tendency to those signals to be in quadrature, but it's not the case all the time. Like, for example, here they are more in phase opposition. So this leads to lower values of coherence, but still significant coherence. So what we've been doing is to record hand kinematics with an accelerometer, attaching to the tip of the index finger, to the top of the index finger, and then ask subjects to make some repetitive flexion extensions, difficult to say, like this, at frequency of about 3 Hertz, with touching and without touching the fingers. We recorded MEG with an Electa device, similar to the one you have now, and applied coherence analysis between brain signals and acceleration signals. So what we showed, so you can see here, the power spectrum of the acceleration and the coherence spectrum, so between 0 and 1. And the dashed line corresponds to no touch condition, and the full line corresponds to touch condition. So coherence always peaked at the same frequency as hand movement. So at movement frequency, which will be noted now F0, and its first harmonic F1. So in all our 10 subjects, we had this coherence at F0 and F1 in touch and not touch conditions. So we decided to go for some source localization at the group level, and we identified a large sensorimotor network with the dominant area, which was the primary somatosensory sensorimotor cortex, contralateral to hand movement.
Sorry, what's the small brain?
So this one is the original coherence map. But then from this, it's quite difficult to extract those local maximum. And this one here is just some enhanced visualization of local maximum. Kind of, yeah.
So it's not just a thresholding?
No, it's not just a thresholding. This one is just a thresholding, yes. Yeah, exactly. But then when you see a coherence map, or whatever map you obtain with source reconstruction, then one should just focalize on those local maximum, because the rest is just some source leakage from the local maximum. But this previous study couldn't tell apart the contribution of motor comment, proprioceptive feedback, or tactile input. So we decided to design a study that would kind of give some more information as to whether the coherence phenomenon we saw was due to proprioceptive feedback, motor comment, or tactile input. So we had subjects doing some repetitive finger tapping, and also we used a passive condition where we just moved the index finger of the subject with an allium stick at the same frequency. And we had also touch and no touch conditions. So the rationale is that when you perform passive movements, you don't send any motor comment, but the proprioceptive feedback is preserved. So we kind of, by comparing active and passive movements, then we expected to be able to say, okay, there is the same level of coherence in the two conditions, so it's mainly due to proprioceptive feedback. But if we observe no coherence with passive condition, then it's good sign that it's due to motor comment sent to the muscles. Just a quick remark, it's that it's not because it's passive movement that we do not observe any M1 activity, because it's quite well known that proprioceptive feedback goes to S1, or there might be even some direct connection to M1, and then the information from S1 and S2, even S2, is directly transferred to M1 with quite short latency. So first we made sure that subjects did not produce any muscle contraction during the passive movements. So what you can see here is that muscle activity during passive condition is non-significant compared to a resting condition, and this activity is highly significant during active conditions. And these are just two different muscles involved in this finger tapping task. We compared as well the location of the corticokinematic coherence in all those conditions and couldn't show any difference in localization. But the localization was clearly different from a simple pneumatic stimulation. And then comparing the strength of the corticokinematic coherence, we showed no effect of tactile afferences. So touch and no touch conditions led to the same amount of corticokinematic coherence. And comparing active and passive conditions, we showed that at F0, so at movement frequency, coherence amplitude was similar, whereas at F1, so movement first harmonic, coherence was even higher during passive condition. So kind of showing that this corticokinematic coherence is mainly due to proprioceptive feedback. And so now we even have some more direct arguments using renormalized partial directed coherence that shows that the flow of information is really from periphery to the brain.
So the area that you show there, that's very similar of all conditions. What area would you say is that?
Primary sensorimotor cortex.
Primary sensorimotor.
Well, actually, we did some testing, kind of to define some kind of volume of confidence, but some area of confidence of this activation. And this area of confidence was mainly overlapping the motor and sensory cortex. With MEG, it's always very difficult to tell apart whether it's coming from sensory or motor side, because it's really close. It's really.
But if you look at the sensory evolved fields, then those seem to be at least anterior to your results, right?
Yeah, a little bit, but they were a little bit deeper in the sulcus as well. It's like, yeah.
But forced choice, this would be sensory.
No, I would stick to sensory motor, sorry. But yeah. Yeah, so just suggesting that CKC is mainly driven by proprioceptive feedback. So we wanted to use as well this corticokinematic coherence phenomenon to probe the mirroring functions in the human brain. So first, let me recall what is mirror neurons. It's neurons that activate when performing an action or when observing or hearing this action performed by a third person. So these neurons have been first discovered in the monkey's cortex, in the ventral premotor cortex, and inferior parietal lobule. And since then, there have been a lot of studies about mirror neurons and inferring the role of those mirror neurons, because they could serve social interactions, because they kind of give some inner representation of other people's actions and could lead naturally to understanding of other people's actions. It's been debated whether M1 in monkey contains some mirror neurons. At least some people show that some neurons in M1 cortex have some mirroring properties, but people do not really speak about mirror neurons in M1. In human, this set of brain areas displaying mirroring properties is thought to be wider, like containing more areas than in the monkey's brain. But the difficult point with human is that it's difficult to come and put electrodes and record directly neurons. So one usually go for non-invasive imaging such as fMRI or MEG. And then we can just show that some brain area activate in response to movement observation and execution. But we cannot say that it's mirror neurons, because it could be that there are two different populations of neurons, one responding to movement observation and the other to movement execution. So a careful term is to use action observation network. So we wanted to probe action observation network with our corticokinematic coherence approach. So we had subjects observing repetitive movements of an experimenter. So what we showed is that the main area activated is visual cortex. But then we also had some local maximum of coherence in the left and right cerebellum, though it's still difficult to say whether it's due to conduction or leakage of this activity due to the method used, or whether it's really in the cerebellum. But we also observed activity in the primary motor cortex bilaterally. So kind of suggesting that in humans as well, the primary motor cortex is activated. So now we'll continue with the clinical application of this corticokinematic coherence. So we've been showing that coherence mainly comes from the primary sensorimotor cortex contralateral to hand movement. So this could be a useful tool in clinical setting, because, for example, in subjects, it's not so difficult to localize the central sulcus from a neuroanatomical point of view. So you have many landmarks. You have this hand knob, and then you can observe also this. I know you can observe it. I'm not really expert with that, but you have different landmarks that allow you to identify this primary motor cortex. The situation is so, on the other hand, much more complex in patients when they have some big lesion like this one, kind of disturbing the local neuroanatomy. So you cannot really say, well, in this patient, maybe one could say, okay, the central sulcus is there, and I think it was the case. But still, it's really informative to have some localizer telling you, okay, this is really the central sulcus, and telling the neurosurgeon, be really careful with this patient. The central sulcus is there, so hand area is there. If you go and remove this big tumor, try not to remove too much of the cortex in this area. So we showed that corticokinematic coherence can be used to map primary motor cortex in population of 10 subjects. So the dipole, the magnetic field pattern was quite specific to motor cortex, and we could localize it quite accurately in all our subjects. So kind of suggesting that CKC, or corticokinematic coherence, is a nice tool to map this sensorimotor cortex. But yeah, let's just go there. So the idea, as we just summarized here, basically, is that the more information you have about the localization of this primary sensorimotor area, the better, basically. So we decided to check whether a scheme that would integrate many functional indicators could bring some more information. So we had patients, 4 patients and 10 subjects coming and doing a multimodal mapping of their sensorimotor brain functions using corticomuscular coherence. So I guess you're all familiar with, are you, corticomuscular coherence? I don't, because I guess my time is already coming. And then mapping the blocking of the mu rhythm. So the mu rhythm is the sensory motor rhythm that kind of reflects the activation state of the sensory motor cortex. The less, like the lower power of mu rhythm reflecting activation and the higher power reflecting some inhibition. Although the role of mu rhythm is still not perfectly known, but it's some current hypothesis. So we mapped the blocking of the mu rhythm at 10 and 20 Hertz, so giving 2 functional indicators. And then the median nerve stimulation, because this has been used extensively to map the central sulcus. And we combine this also with fMRI motor paradigm. So we had 7 functional indicators of this primary sensory motor cortex. So 7 coordinates that we combined with principal component analysis. So we just derived an ellipsoid that would fit the cloud of functional indicators. And then the major axis and the volume of this ellipsoid kind of give some information about how those functional indicators are spread. So here are the results, not so easy to see because there are many subjects and patients. But yeah. So in all participants, we had from 5 to 7 functional indicators that were passing our rejection criteria. And the center of this ellipsoid always fitted quite nicely with the central sulcus, and so some primary sensory motor area at the hand area, as we could define it from anatomical landmarks. And then maybe some interesting remark is that in 80% of subjects, the dipoles or the functional indicators was less than 10 millimeters from the center of the ellipsoid, whereas it was only 50% of those functional indicators in patients, kind of suggesting that tumor induced some deformation and some spreading of motor functions. I go to conclusions. Last slide.
Sorry, is there one, could you say a little bit more qualitatively then also what method was maybe best used or had the best results for the patients versus control?
Well, I really think that's, well, first, no wonder median nerve stimulation quite robustly pinpointed the central sulcus. And then the other methods were more or less equivalent. Like in subjects, for sure, fMRI worked quite well, but in some patients, we had also, I guess in 1 or 2 patients, the fMRI localizer was not especially very good, at least matching with the other. But I guess it's more the combination of all those methods that kind of give a global picture. Because, well, CMC, some people might lack it, simply because they don't have CMC. Cortico-kinematic coherence, if patients have some implant or if they kind of struggle to make movement, then it's quite difficult to record it. Then fMRI, when you have some big tumor, it's quite difficult to, well, there are a lot of problems related to the perturbation of neurovascular coupling related to that. I don't see any problem with CEF, with median nerve stimulation. And mu rhythm, yeah, mu rhythm is less easy to localize, let's say, because it requires going through beamformer.
Yeah, and that even if there's one patient that benefits, right?
Yes.
It's of a big benefit.
Yeah. Well, qualitatively, it was really nice for medical doctors to have all this information, like confirming each other. So just to go through some conclusions about all those studies. So about the possible origins of corticokinematic coherence, it could be due to movement encoding, proprioceptive feedback, and tactile input. And we kind of showed that it was mainly due to proprioceptive feedback from hands, kinematics, so 2 brain signals. About the possible origins of corticokinematic coherence during observation, in the context of moment observation. So the first possible hypothesis is that it's due to encoding of observed kinematics. So it would kind of reflect some mirroring properties of M1 neurons. But then the activation could also reflect some inhibition of the motor cortex, so as to prevent involuntary imitation. And actually, some disturbance or some malfunctioning of those mechanisms to prevent automatic imitation leads to some neurological disorders such as echopraxia. I don't know if you have heard of this one. Rita, he likes to mention it. And then finally, it could also be due to mirroring of the somatosensory consequences of the movement once observed. And this kind of hypothesis is pushed forward by the team of Cazzola. She's working a lot about somatosensory mirroring. And then to conclude about the clinical applications, so corticokinematic coherence has high success rate and could be a nice functional indicator to include in a clinical setting. But then one must be really careful that patients are artifact-free or meter-free, that they do not produce too strong artifacts that will anyway render the analysis impossible with corticokinematic coherence. So thank you for coming and for your attention, and I also thank the lab where I did my PhD in Brussels, and my supervisor, especially Xavier de Thiège, was been helping me a lot with all those neurophysiological concepts. And also the team in Espoo, Rita Rivek Koljusmäki and Harri Pitulainen, who I'm working a lot with nowadays. Thank you.
Thank you.